Drop a like and do share. Leave your comments and do not forget to subscribe for more videos.
Hello students, my name is Niyati Said and thanks for watching Wikipedia word videos. My topic for the presentation is 6th section of the chapter Cell, the basic unit of life. In this section of the video, we will discuss about the nucleus, which is a cell organelle present in eukaryotic cells. But in prokaryotic cells, it is present in the form of nucleoid, and not in the form of nucleus. So what is nucleus, students? Nucleus is like a brain of your cells. The word nucleus can refer to the center of an atom or control center of a cell. See, it is present in the center. This is nucleus, and it is present in the center of the cell. So the word nucleus can refer to the center of an atom and control center of a cell. You have over 200 different types of cells in your body. Some can be really thin and long, like nerve cells that run all the way from your spinal cord to your foot, students. Some, like a fat cell, have more rounded shape. No matter what your cells look like, there is an important thing they all have in common, and that is nucleus, students. So this is the typical diagram of a cell where cell organelles are present, such as mitochondria. See, this is mitochondria. Golgi bodies. See, these dots are Golgi bodies. These are ribosomes. This is endoplasmic reticulum, which is present in the form of mesh-like structure or in the form of reticulum, and this is Golgi apparatus. And this is your nucleus, students. So nucleus is a control center that is found inside all of your cells at some point during their life. A nucleus is responsible for determining and controlling what a cell looks like and what it does. So the nucleus was first described by Robert Brown in year 1831. And the material of the nucleus, which was stained by basic dye, was given the name chromatin by Fleming. Fleming was the name of the scientist. Normally, a cell has only one nucleus. That means a cell has one control center. Some mature cells, they lack nucleus, students. For example, mammalian RBC and sieve tube cells of vascular plants. They lack nucleus because they are mature cells. The interface nucleus, or you can say what is interface? So it is a nucleus of a cell when it is not dividing. So the interface nucleus, it has highly extended and elaborate nucleoprotein fibers, which we call it as chromatin, students. So this is your chromatin. This is chromatin, which is present in nucleus. So what is chromatin, students? It is highly extended and elaborate nucleoprotein fibers. So these are fibers, and they are nucleoprotein in nature. And what is nucleoli, students? Nucleoli is a nuclear matrix, and one or more spherical bodies are called as nucleoli. Nucleoli singular form is known as nucleolus. So this is nucleolus. What it is? Neuromatrix, and one or more spherical body is called as nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between, is called as perinuclear space. See, students, a nucleus, it has two membranes, two parallel membranes, and the space between two parallel membranes is called as perinuclear space. That forms a barrier between material present inside the nucleus and that of the cytoplasm. So the interface nucleus contains nuclear envelope, nuclear matrix, which we call it as nucleoplasm, and chromatin, which are highly extended fibers, elaborate nucleoprotein fibers, which we call it as chromatin, and nucleolus, which is a nuclear matrix and one or more spherical body, which we call it as nucleolus. As I told you that nucleus, it has two membrane, or it is a double layered cell organelle which is present in eukaryotic cell. So it is a double layered membrane with a space between two membrane is called as perinuclear space. So this is perinuclear space, which is present between the two parallel layers of nucleus. See, this is the outer cell membrane and this is the inner cell membrane present in the nucleus. Okay, and the space between these two parallel layer membrane is called as perinuclear space. It is a barrier between the material inside the nucleus. See, this is the inside of the nucleus and outside the nucleus is cytoplasm. So it creates a barrier between the materials in the nucleus and the cytoplasm. Okay, now outer membrane, which is this one, it usually is continuous with the ER, that is endoplasmic reticulum. I have already taught you endoplasmic reticulum, so for better understanding you can refer to previous presentation. Okay, so this outer layer is in continuous with the endoplasmic reticulum. See, these are endoplasmic reticulum and these small dots are ribosome, which are protein factories where protein is synthesized. Okay, and this is nuclear envelope. It has minute pores, as you can see, which is formed by the fusion of its two membranes. See, these are the pores which are present in the nuclear envelope, which is formed because of the fusion of these two layers of nucleus. Okay, and these are the passages for the movement of RNA and protein between nucleus and cytoplasm. That means these are the small pores where RNA and protein can be transferred from nucleus to the outside world, that is cytoplasm. Okay, so this white portion is cytoplasm. So from these small minute pores, RNA and protein from inside of the nucleus can be transported from inside to the outside, that is cytoplasm. Okay, now what is chromatin, students? Chromatin, these are a network of a nucleoprotein fibers. It contains DNA and basic protein, that we call it as histones, and non-histone proteins and RNA. Okay, so chromatin contains DNA and basic proteins, which we call it as histone, and non-histone proteins and RNA. Okay, so this is your protein, chromatin, that is nucleoprotein fibers. Okay, network of nucleoprotein fibers rather. During cell division, chromatin they condense to form chromosome. What is chromosome, students? The nucleus of a cell, it contains majority of cell genetic material in the form of multiple linear DNA molecules. Okay, we very well know that. So these DNA molecules, they are organized into structure which we call it as chromosomes. In a cell, they are organized in DNA protein complex, which we call it as chromatin. So this is our chromatin, where DNA and protein complex is organized to form chromatin. During cell division, chromatin forms well-defined chromosomes. The genes within the chromosome, they consist of the cell's nuclear genome. Okay. Now come to the nucleolus part. Nucleolus is not surrounded by membrane. See, this is nucleolus, okay, and this is nucleus. So nucleolus is present inside, or it is present in the center of the nucleus. So what does nucleolus do? It is not surrounded by a membrane and it is densely stained structure which is found in nucleus. The nucleolus are formed around the nuclear organizer region. And what does this nucleolus do, students? It synthesizes and assembles ribosomes and rRNA, that is ribosomal rRNA. The number of the nucleoli is different from species to species, but within a species, the number is fixed of nucleolus. During cell division, nucleolus disappears. It just disappears from the nucleus during cell division. Studies have also shown that nucleolus may be involved in cellular aging and senescence. Okay. So what is nucleolus? Now it is clear to all of you now that it is one or more non-membranous spherical body. Okay. And it is continuous with the nuclear plasm. Nuclear plasm means cytoplasm of nucleus, is known as nucleoplasm. And it is site for ribosomal RNA synthesis, that is rRNA synthesis. Now this is chromosome. I have already taught you what is chromosome. You may recall that interface nucleus has a loose and indistinct network of nucleoprotein fiber, which we call it as chromatin. I just taught you. Okay, but during different stages of cell division, cells, they show structured chromosome in place of the nucleus. So this is chromosome. Chromatin contains DNA and some basic protein, which we call it as histones and non-histone proteins, and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 chromosomes. Every chromosome, it has essentially a primary constriction, which we call it as centromere. So this is our centromere. This is primary constriction. Okay, on the sides of which disc-shaped structure, which we call it as kinetochores. See, these are the kinetochores, which are disc-shaped structures are present. Okay, and these are the mitotic spindle microtubules, which helps in the segregation of these two sister chromatids during anaphase. Okay, anaphase, I'll teach you what it is. But for the timing, just know that this is the typical diagram of a chromosome. It has a primary constriction, which we call it as centromere, on side of which disc-shaped structure, which we call it as kinetochore. See, this is disc-shaped structure is present, which we call it as kinetochores are present. And these two are sister chromatids. Okay. Every chromosome essentially has a primary constriction, which we call it as constriction, and on the side of this, disc-shaped structures are present that we call it as kinetochores. Okay. Now, based on the position of a centromere, chromosomes are of 4 types. Which are those, students? First is metacentric chromosome. Middle centromere forming 2 equal arms of the chromosome. That means when centromere is present at the center of 2 equal arms of the chromosome, then that is metacentric chromosome. Now, what is submetacentric chromosome? When a centromere, see, this is the centromere, which is nearer to one end, forming one shorter arm and one longer arm. As you can see that this is submetacentric chromosome, where a centromere is present near to one end. See, this end is very close to this centromere. So, centromere is nearer to one end, forming one shorter arm, and this is one longer arm. So, this position is called as submetacentric chromosome. Then comes acrocentric chromosome. What is that? Centromere is very close to its end, forming one very short and one very long arm. See, this is acrocentric chromosome. This is chromosome which is present near to its end, forming a very short arm and very long arm on the other side. Then comes telocentric chromosome. Telocentric chromosome means terminal centromere. That means if you just remove these 2 small arms, then you will see that this is the centromere and this is the only arm present below it. So this is telocentric chromosome, which has terminal centromere. Now, some chromosome, they have non-staining secondary constriction at a constant location, which we call it as satellite. So this is satellite, which is present in few chromosome, and this is non-staining secondary constriction. See, this was the first or primary constriction, which we call it as centromere, and this is the secondary constriction, which is non-staining secondary constriction and which is present at the constant location, which we call it as satellite. Okay, but it is present only in few chromosomes. Now comes microbodies, students. What is that? Many membrane-bound minute vesicles are called as microbodies that contain various enzymes which are present in both animal and plant cell, students. Okay? So this is microbody. These are membrane-bound minute vesicles that contain various enzymes. See, this is the lipid bilayer which is present outside the cell. This is also known as plasma membrane. Okay, and please note that both plant and animal cell, they contain very small granular bodies in their protoplasm, which is surrounded by single unit membrane. Such structures, they are different from lysosomal vesicles because of their specific physiological properties. They are called microbodies. Okay, and students, please note that their size varies from 0.6 millimeter to 1.5 millimeter. The number of the microbodies also varies from 20 to 100 per cell. And please note that in germinating seeds, microbodies increase in their number considerably. The content of such structures may be amorphous, may be crystalline or fibrillar in nature. And however, the enzymatic contents in them may vary. Based on their functional properties, they can be classified into peroxisome and glyoxysome. Okay, based on their functional properties. Okay, so this is about the microbody, that they are membrane bound minute vesicles which are present in both plant and animal cells, and they contain various enzymes. And based on their functional properties, they are classified into peroxisomes and glyoxysome. I just give you what is peroxisome and glyoxysome. So peroxisome is characterized by peroxidase activity. Okay, and they are more or less dense spherical bodies which is bounded by single unit membrane, and it is almost present in every species of plant kingdom. But among animals, only higher vertebrates contain such structures, that is peroxisome. Okay, and most important, the enzyme component of peroxisome is peroxidase or catalase or both. Now come to the glyoxysome, which is another category of microbody students. They are distinct from peroxisome because of the different functions. They are found in fungi and other higher plants. They are storage organs which are rich in oil globules that contain greater number of glyoxysomes. Okay. And please note that glyoxysome, they contain a host of enzyme which is responsible for beta oxidation of fatty acid. Thus, it has all the enzymes which are necessary for fatty acid oxidation as well as glyoxylate cycle and gluconeogenesis. Okay, so this was all about nucleus and microbodies. In my next section of the presentation, I will teach you about the cell membrane, that is plasma membrane, in detail. Okay, so till then, stay tuned and keep watching Edupedia word videos. Thank you.